guys might know. Okay, let's go here. So what we're going to talk about today is chapter 8, cellular reproduction. So we're basically going to take into a scenario where let's say that you are just walking along the sidewalk and you trip over a rock and you totally scrubbed up your knee, right? And you have to go get a band-aid and you have to put it on a band-aid. But that knee, that scab that you or a wound that you caused is going to eventually reproduce, right? We're going to get cell reproduction where we have the cells duplicating to repair it, and it's going to end up with healing itself, right? That when we scrape ourselves, our wounds don't just stay open, like our skin doesn't just stay open, it's going to actually repair itself. And so how does it undergo that? That's kind of what we're going to talk about today. So first, let's just kind of think about how we start off with just one cell, right? You have the sperm and the egg, they combine, they fertilize, they form a zygote. How does that one cell, that zygote, lead to us? Well, that one zygote is going to divide and become 2 cells, and then those 2 cells will divide and become 4 cells, and then those 4 cells will divide, become 16 cells, and so on, so on. So that in the end, you end up with being made of trillions of cells, right? And why do we do the cellular reproduction? Well, we do cellular reproduction so that our zygote can develop, so that that one cell can develop to be a being later, to do tissue repair. So like I said, you scratch up your knees and now you need to repair yourself, right? So that's another situation. And just to grow, how do you go from being a tiny baby to eventually being a toddler to a child to an adult? It's because you're constantly growing, because your cells are constantly dividing, and we should divide enough that we end up being able to grow. So there's 2 types of reproductions that we should think of, right? So we're going to focus today on asexual reproduction. The next lecture will focus on sexual reproduction. So asexual reproduction is the fact that the child cell that's forming is from the parent cell. So we don't need any sperm or egg. We're just dividing over and over the same cell. It's dividing. So the child cell is going to be identical to the parent cell. In sexual reproduction, we're combining genetic information from 2 separate individuals. So 2 separate individuals can come together. So this would be like the sperm and the egg come together and they form the zygote. Cell division has 2 parts. The first part is the DNA has to duplicate, and not just DNA, but all the other things that's in the cell, in the cytoplasm, so organelles, microtubules, like just all that information is going to have to duplicate. So you end up with this kind of big piece of cell for a while the DNA is duplicating. And then eventually, once it has a signal that division's done, right, it's going to actually, or that the cell's ready for division. It gives the signal the cell's ready for division. It will actually pinch down and do what's called binary division. And this is when we kind of basically form a cleavage, and we're going to end with 2 daughter cells. And so during cell division, what mainly happens? Well, the DNA is going to replicate because we want to replicate the DNA. The DNA is super important, has all that genetic information, all that blueprint. And we want to give a full set to each one of our daughter cells. The DNA is going to be packaged up with DNA, and it's going to wrap around these proteins. These proteins are called histones. So DNA is going to wrap around, and this is so that we can divide that information easier between the 2 daughter cells. So you know how when you look at what we consider a classically chromosome, you can see that has a very condensed structure. Normally in the nucleus, the DNA is kind of a little bit more loose, like it's a little bit more like an unwound ball of yarn, I want to say, kind of loose. But when we're ready to distribute them, they start wrapping around these proteins really tightly, because we want to make it so that we can ensure that the genetic information is actually separated appropriately to the daughter cells. And so sometimes, normally we're like a thing of spaghetti, I want to say. This looks like a little thing spaghetti. Then we're gonna wrap all of our DNA information around these guys. And at the end, we end up with something that's very dense looking, like so one chromosome replicate into two chromosomes. Wait, sorry. DNA spaghetti will be packaged into a tight chromosome. Chromosome is going to replicate, with two chromosomes. This is two sister chromatids. These sister chromatids are pretty much identical to one another. And then we're going to separate out the sister chromatid, one to each of the daughter chromosome, so that in the end, we end up with going from a parent to two child that has the exact same genetic information. So the DNA, like I mentioned, is going to wrap around these proteins. And so let's take a look at how that actually happens so we can see how the DNA wraps around these proteins to form what we see as a chromosome.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, I think it's always a cool video to kind of see how it actually happens. It's pretty amazing that there's like signals in our body that can actually condense these guys into the chromosome. So you can kind of see this is kind of what it looks like before it really starts moving on top of one another. So you end up with all these little nucleosome here. This is the loose DNA we wrap around these histones. Okay, so we're going to look at the cell cycle. So we're looking at mitosis here. Most of the times, our cell is not dividing, and we're in what's called interphase. So it's just kind of hanging out there. And when we get the signal, right, we're going to grow. So when we're growing, our organelles will double, the internal cytoplasm will double. We're going to go ahead and go under S. So G1 is growing, so G for grow. Then we're going to go under S, where we're replicating. So this is S for synthesis. We're going to replicate our DNA right here. Then we're going to move into what's called G2. G2 is where we actually find chromatin, which is when the DNA is kind of starting to wrap around. The histone, but it's still not super tightly wrapped around yet. It's kind of wrapping around, and the cell is growing even more because the cell knows that, okay, we're going to undergo some cell division. So we're gonna get ready to divide. So it's gonna grow more, and then we actually then enter mitosis. Mitosis, what you're gonna have to do is you're gonna see the DNA wrap up even more tightly into the chromosome structure, and then we're gonna go into metaphase, which is when the chromosomes are lining up in the middle because they're getting ready to divide, right? Remember, the whole point of mitosis is to make two daughter cells from the parent cell. So we're going to line up our chromosomes because we're going to want to split the chromosome into two daughter cells. So they line up, and then we go into anaphase, which stands for away because they're going to pull away we're going to telophase where they signal that, okay, now we have it ready for us to divide. We have our DNA information and all the cell information kind of on two opposite ends. We'll see this in a little bit when we look at the actual visualization. But we're going to line them up on both sides, and then we're going to go under cytokinesis. And cytokinesis is when we actually divide up. In the center, we form the ferrule, and now we end up with two daughter cells that are identical to the parent cell. So again, to reiterate, G1 is our first row or gap, right? It can be either one, but I basically think of them both as like growing. It's a time in which we're not dividing. So cell one, we're growing, where the organelles have doubled. We haven't decided if we're going to divide or not at this point. So if we don't divide, we go to GO, which means, okay, never mind. Let's just hang out here and chill. If we get the signal we are going to divide, then that's when we actually undergo S for synthesis, when we're actually going to replicate our DNA. So now the chromosome each replicates, that we end up with two sister chromatids attached by the centromere for each of the homologous chromosomes. Does that make sense? We have a pair of homologous chromosomes. They are going to replicate. So at this point, it's just one chromosome from one mom, one from dad. But we have a pair of them. They're going to replicate. Each one of the homologous chromosomes is going to have two sister chromatids. Those sister chromatids. So now we have two homologous, each with two sister chromatids. We're going to enter into G2. We're going to synthesize some more stuff that we're going to need for division. So we're going to synthesize some proteins and such. Then we're going to actually enter into mitosis. Mitosis is when we actually have the chromosomes up, and we're going to separate them, and the end result is two child or two daughter cells. Okay, so what phase did the organelles double? Remember, it's going to have to not be mitosis because that's we actually undergo division, right? It's going to be G1. It's in this phase that the organelles are doubling. Now we have to decide, okay, do we want to divide or not? If we do divide, DNA is going to replicate. If we don't divide, it goes into G0, which is kind of like a rest state. So basically, it's just like, okay, well, whatever. I'm just going to chill here. And if we have too much organelles at that point, our body recycles. It breaks things down. When it doesn't get the right signal, it's just it breaks down. When it needs it, it rebuilds it back up. So in the mitosis cell itself, that's when we're actually gonna have division, cell division. Because in mitosis, we already have the DNA replicate, and we want to actually separate out the DNA, but we also want to separate all that organelles and cytoplasm that we duplicate. We're also going to separate that. So in the end, we have two daughter cells that are pretty much identical to the parent cell. Okay, so we want to produce two daughter cells that are the same as the parent. So it should have the same information within its cell as far as the organelles, the cytoplasm. It should also have the same number of DNA, right? Genetically wise, it should be exactly identical to the parent.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing two copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has four stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of two genetically identical daughter cells.
Yay, so now we have 2 daughter cells. Okay, oops, see. Okay, so microtubules are basically just different proteins. That's where the spindles are formed, and it's when they get the signal that we want to undergo cell division that they form. And so there are things that's going to pull the sister chromatid, and they will actually, as they're shortening, they're kind of breaking down. There's a whole process. We won't go into it. All you need to know is that the microtubule is going to hang on to the sister chromatids, and they're going to split them up. So that's why in the end, you end up with daughter cells that are identical to your parent cell, because you duplicate them, duplicate everything, and then you're just splitting into 2. And so here are the different chromosomes. So you end up with the parent cell having 23 chromosomes, 22 of them are autosome. You'll learn this more in the next lecture. The autosome are the non-sex chromosome. And then in humans, we have either an X or Y as our sex chromosome. So we have 23 total chromosomes. You can see them kind of pretty here, right? 23 version. We take when we're undergoing mitosis, we're taking a 23 chromosome, replicating it all so that we end up with 46 chromosomes. And then we're going to, sorry, let me re-say that. We have 23 chromosomes, but we are diploid. So we technically have 46 because we have a set of 23 from mom, a set of 23 from dad. We're going to duplicate that all so that we have 92. That's what I meant to say. 92 sets. That 92 is going to be divided again. So then in the end, we end up with 46 in each of the daughter cells. And that's once we have the cell division and the correct number of chromosomes in there, then we're done with mitosis. So we went from diploid, which means that we have 2 copies of each chromosome, made ourselves into a 4N, but that 4N was very temporary because we went back down to 2N and 2N. So 2 daughter cells that are identical to the parent cell. And we're done with mitosis. Next time we will talk about meiosis, which is going to occur only in the sex cell. Remember, mitosis is happening everywhere. Mitosis is happening for you to grow. Mitosis is happening for you to repair injury. Mitosis is happening for you to replace cells that are just getting old and just need to be replaced. Meiosis, which is what we'll talk about next time, only occurs in our actual sex chromosome. Okay, so that's all we have for this chapter. I will see you guys next time in class. All right, bye everyone. Okay.